Hi, my name is Cesar Ayala, and I am a graduate student at the National University of Mexico. And today I'm going to talk about a tool that we've called MetaCore. Unique core sequences from metagenomic samples for microbiome wide association studies. I must mention that this work is part of my master's degree thesis. So, what is a microbiome wide association study and what's for short? They are based on the GWAS methodologies that tries to understand the intricate relationship between genetic variations and observable traits. This was known as the common disease, common variant hypothesis. Now, when applying association methods to microbial communities, the challenge increased as the taxonomic, the gene content, or the diversity metrics information could be tested, and indeed has been tested mostly in relation to human phenotypes. However, in our lab, we are trying to apply MWAS methodologies to the study of environmental phenotypes, also under the hypothesis of common phenotype, common variant. In our case, the phenotype under study is the degradation of textile dyes, which are of high concern due to its toxicity and contamination potential in the environment and potential risks to human health. In this case, as there are no longer hosts, we are trying to answer the question, which are the genetic variants responsible for the phenotype in such diverse microbiomes? Therefore, to reduce the MWAS analysis complexity, we have developed a bioinformatic tool that we call MetaCore. This tool considers each microbial community as an individual genomic entity, aiming to reduce the complexity and dimensional structure of microbial shotgun sequencing. How it does it? Let's consider a case control association study, where each microbiome represents an individual sample. Therefore, we will have two groups with n number of samples. So the goal is to find the common genetic information within the phenotype positive samples and then test it out with the Pfizer linear mixer model implementation. Therefore, in a first step, the pipeline extracts all the DNA sequences shared with a fixed identity percentage between metagenomic assemblies in a cyclic and alignment-free manner using MashMap. For this case, we've chosen 85% identity, but that can be changed by the user. In each cycle, a subfile containing the common sequences between the reference and the sample is created. Then the process repeats itself with all the positive samples. In the step 2, all the subfiles that we previously created are concatenated in a unique reference to extract from it all the common sequences that are present in the metagenome assembles of the negative samples. So this pipeline will output subfiles that we've called map and mapneg, whether they come from the positive or the negative samples. MetaCore was tested with 84 samples, 23 positive for the textile dye degradation phenotype and 61 negatives for the phenotype. MetaCore allows us to reduce the total amount of variants in almost a 74% when compared to the total amount of variants in the microbiomes. Additionally, in a linear mixed model, with variant frequency of 20% or more, it allowed to test almost 11 times more variants in the model. The latter allow us to achieve a decent adjustment of the model and a reduction of the p-value inflation as seen in this quantile plot without the inclusion of any additional covariate in the MWAS MetaCore strategy. Finally, in the form of the Bright-Curtis diversity metric, the use of MetaCore results in a better separation of the groups, as seen by the PCA plots and the ANOSIM static error, which in the case of the MetaCore strategy is almost closer to 1 than when all the information in the microbiomes was used. In summary, full metagenome data increases the dimensionality and the expensiveness of computational resources. We've proposed MetaCore alternative, which reduces the complexity as much as 3 times and increases p-value astringency, which also helps to reduce false positive variance in the MWAS analysis. Performing an in-depth comparison of the MetaCore algorithm with a group of synthetic data will be an additional validation test necessary both to understand the limitation of computations and its applicability in other experimental designs. Finally, I would like to thank publicly to the Programa de Apoyo a los Estudios de Posgrado PAEP de la Universidad Nacional Autónoma de México through the call to carry out short duration national or international academic activities 2024 and to the Consejo Nacional de Humanidades, Ciencias y Tecnología, CONACYT, for the granted scholarship. Thanks, that's all for me.